All right, so welcome to AP Bio, Unit 1, Topic 6. This one's on proteins. So what is a protein? Uh, what is a protein and what is its purpose? Basically, a protein is a biomacromolecule that makes things happen, essentially. And it makes things happen, and its two main purposes are, one, performing chemical reactions. This is really important. Everything that happens in your body, most actions that happen in your body are caused by different forms of proteins performing chemical reactions, and it's very, very crucial to living. Some people would argue that the basis of life is proteins, essentially, because they just do everything. And then the second purpose of proteins is to provide structure to cells. And this comes in many forms, and you'll learn about different forms of proteins that provide structure in future units, Unit 2, especially with passive transport, but we won't focus on that in this video. Zooming out, the golden rule of proteins, per se, not officially, is that shape is everything with regards to proteins. Shape means everything. It's so crucial and so important because the shape of a protein determines its ability to function, and without function, what's the point of a protein if it can't do anything? Going over a brief overview of the structure. We'll get into way more details in a minute. But the monomers of proteins are what's called amino acids. Let me write that in. Which are the building blocks of proteins. And then the polymers are, of course, proteins. Diving into what amino acids are. They are the building blocks of proteins, and this is the basic structure of an amino acid. First, let me get a nice gray color. Right here, we have the central carbon, central C. And it is super crucial because it's the center of each amino acid, and it's what ties everything together. At the bottom, it's connected to a hydrogen. All amino acids have this hydrogen at the bottom. On the right, we have a carboxyl group, and you should know this from a previous video. If you don't, check it out where we went over functional groups. And then on the left, we have the functional group, the amino group, right there. And then at the top is what makes each amino acid special. Right here is the R group, also known as the variable group. And this changes for each amino acid that there is. There are 20 different types of amino acids, which are caused by the fact that there's 20 different R groups. So each R group attached to this other structure at the bottom creates each of the 20 proteins, basically. And different combinations of the 20 amino acids is what makes up proteins. So what is really important to understand is how do we go from a single amino acid to a chain of amino acids that can form a protein. And right here, we have our favorite, dehydration reactions. You guys should be familiar with this from previous videos. If not, go check them out. But dehydration reactions are very crucial for the forming of almost everything. In the case of amino acids, this is how it works. Two amino acids will line up in this orientation, such as that they are next to each other, with the carbons and the hydrogens facing the same direction. And on one side, there will be the carboxyl group of one protein, and on the other side, we have the amino group of another. And what happens is through dehydration reaction, we'll see that the OH group from the carboxyl and the hydrogen, and one of the hydrogens from the amino group, will essentially be taken out and form water. And as a result, the carbon and the nitrogen will be bonded. It is pretty simple. Long chains of these amino acids will be formed through this method. And this is the basis of how singular amino acids can form proteins. For AP Bio specifically, you don't need to memorize each of the 20 amino acids, but you should know how they're categorized. And they are categorized by two things, which is acidity and polarity. In terms of acidity, we have acidic proteins, and we have, amino acids, and then we have basic amino acids, and then there's polar amino acids, and there's non-polar amino acids. And a combination of various amino acids is how we get our shape, because some non-polars will interact with non-polars, and some polars will prefer to interact with polar amino acids, and this is what creates unique shapes. Zooming in on protein structure, or I guess zooming out, scientists like to categorize the protein structure into four levels of structure per protein. The primary structure is the most basic level of the structure of an amino acid, and it's essentially just the string of amino acids. So if we have like an amino acid string, pretend each of these is an amino acid, and this simply on its own is the primary structure of a protein. And this is important because knowing the string of amino acids, which is also known as a polypeptide chain, is super important for understanding the shape and therefore the function of a protein. Next, we have the secondary structure of a protein, and it's important, and this comes in two forms, which is two structures really, the alpha helix and the beta pleated sheet. The alpha helix is a helix helix as in the name, of polypeptide chains, which is held together by these, which are hydrogen bonds. And I'm just going to go fill it in right here. The secondary structure of amino acids of proteins are formed by hydrogen bonds. And this is very crucial because hydrogen bonds are so important, and they allow for this specialized structure in proteins that allow for specialized function. And in alpha helixes, these hydrogen bonds will hold this polypeptide chain together into this helix shape, which allows for a unique shape and therefore function of proteins. I will keep emphasizing that because it's so important. Shape dictates function. Next, we have the beta pleated sheets, which are usually two lines of amino acids with hydrogen bonds holding it together, and these fold almost like an accordion fold. I think it's called. If you imagine like a piece of paper that you fold the opposite direction each time you switch off which direction you fold it. It's sort of like that in terms of structure. Also formed by hydrogen bonds. Next, we have the tertiary structure, which is the structure that in general we're most interested in, which is the 3D shape. And this is a bunch of squiggles, but pretend this represents what a protein may look like, with a ton of alpha helixes over here, or like beta pleated sheets over here. This is just a bad drawing to try and get the idea across. But these are formed by interactions between, you guessed it, R groups. And this is where R groups come into play, especially because the different combination of acidic, basic, polar, and non-polar amino acids, when those R chains on the amino acids interact with each other, they're going to form a specific shape, as non-polar chains tend to like reacting with other non-polar chains, versus polar chains like to interact with polar chains. And the combination of these plus the acidity and the various acid levels of each form a unique structure that forms a unique function from that shape. Finally, we have the quaternary structure, and this is formed by interactions between different individual, you guessed it, polypeptide chains. And this is formed when we have multiple chains, you could say various proteins, but that isn't exactly how it works. Various different chains of amino acids, which when put together and interacting with each other, rather than within the amino acid chain, it forms a new protein in which all the chains react or go together and react with each other to form the proper function of the protein. Finally, denatured proteins is the last topic of this video, and this is very important. And denatured proteins are essentially proteins that lose their shape, therefore they lose their function. Again, golden rule, shape is everything. And how? There's 3 main ways in which proteins denature over time, or not over time. It could be in an instance. The first is pH, which is the pH in general. The more acid or the more basic, actually, I have an H+, but it could be an OH-. The more acid or the more basic, or basically the extremes of the acidity spectrum, are bad for protein health in general. Each protein has its own various spot on the scale of 0 to 14 of 0 to 14 that they thrive the most at, but in general, a generalized rule is sway too far either way, and it means death for the protein. And this is because if you can imagine the structure of amino acids, they have all these different like R chains maybe going out, and pretend this is like the main chain, and then we have like all these things going out. And if an H+ gets stuck in here and interacts, it could squeeze its way in between. And then because H+ is just a proton, it most likely won't want to react with, let's say, negatively polar R groups, and that may throw off the entire structure. Just one amino acid not working as intended can completely denature a protein, which is scary. Proteins are in general pretty fragile. Same thing with OH groups. They also can interact and therefore cause different problems and denature the various proteins. The next thing, as you just saw, is salinity, and it's very similar to pH in the fact that excess salt can cause a change in protein because the Na pluses and the Cl minuses will interact with various things because of their charge, mainly polar stuff, and that can cause essentially a protein to change shape and denature. Finally, heat is very important. Too much heat can essentially cause a denatured protein, and this is because, and also proteins when they're too cold also don't work as properly functioned. But heat denatures proteins because heat is essentially energy. At the end of the day, heat is energy. It's the vibration of molecules, and when there's too much heat, there's too much vibrations, and that can mess with the hydrogen bonding, especially in proteins, and cause the hydrogen bonds to break or to reform in different ways that isn't expected, and that can lead to very devastating problems for the protein and cause it to denature. Anyways, that's it for topic 6 of the AP Bio curriculum of Unit 1. Please check us out for more AP Bio help. Anyways, thank you.